Okay. Well, thank you very much, and thank you everyone for coming out, even though schools are 2 hours delayed or closed, and I'm sure there's some last-minute things you had to do and able to get here. I don't know how many of you know Tony Lockenbrook. He gave a talk here once in biostatistics, and he said, "Well, it works now." Oh, right. He said, "Once now that you know that I own a jacket, I can take it off." Also, I just found out we're not being videoed, so there's another reason not to have it. Well, I think a bunch of you are here today because of this stepped wedge business. So we'll spend some time talking about this particular design because the whole trial was a stepped wedge trial. And we'll go through an example here, which is going to be from start to end our only example, which is the study that many of the people here have been working on for years, the tuberculosis trial in Brazil. I have about 10 slides on the randomization of it, but I'm going to go through that very quickly because I know this is being made available on the website later. So when I breeze through that, you can always find more if you want. And mainly I'll spend time on a lot of the interpretation of the results and how we think about the data in this particular type of a trial. So here's the site of the trial. You can see right off why we did it there. It's absolutely beautiful. And it takes a team to raise a trial, and there are at least two, and very large teams here, and we've got a bunch of folks here who are on it, Jonathan and Sylvia at least. I'm not sure who else is here. And I can actually talk about the results today because this was just published a few months ago. So we'll give you some of the actual results, which I haven't been able to do previously. Hate to scoop yourself, after all. And this is mainly funded by Gates through the CREATE consortium. So there's a lot of names for this design. Okay, one-way crossover, that's sort of the statistical way to think about it. Phased implementation, pipeline, multiple baseline. And there's been some people say stepped wedge, some say step wedge, some have hyphens, some don't. There's all sorts of ways to do that. Anyone here see any other names for that? For a step wedge trial besides what we have here? I'm going to start collecting them, just like people collect little knickknacks. What was that? Waiting list. Well, it could be, because that's how it works. That's the phased implementation idea. But the basic idea is that, in general, you have a lot of geographic areas. In this case, it wasn't so geographically based, but clinic-based. You start an intervention in one of them, and then you keep it going there, and you go on to the next area, and you keep it going there, and you keep going on to the next area until everyone has the intervention. So that's the idea of the phased implementation side. Probably a good article to get in to read if you're going to start getting into this is this first one. It just published a few years ago in American Journal of Public Health. The second author is the guy who wrote the first book on cluster randomized trials. And there's been a few other review articles talking about different aspects of the trials, and a couple more since, seeing if people adhere to the expanded consort guidelines for cluster randomized trials and all that kind of thing. But this one is good, and I think it's more balanced than some of the other ones. It shows out it has some of the downsides of the stepped wedge that some of people usually don't think about. So, the TRIO study we're talking about today. It's basically a health services type of intervention. On the books, the clinic personnel were supposed to be doing this. They were supposed to be doing testing for TB infection, and then anyone who turns up positive, then they put them on isoniazid prophylaxis. It wasn't happening that much, and the idea was to really train in a much more vigorous way the personnel and make sure everyone is on board here. So it's not like we're testing a completely new drug or anything. Out of the 50-odd clinics in Rio, we had 29 that we got to participate in this trial. These are clinics where people who are living with HIV come to get their drugs, basically. And it's been free in Brazil for years. They really led the way in the world for providing heart. A very impressive operation. The basic design here was that every 2 months we would enter 2 of these clinics in the intervention, which in the intervention is basically a training intervention. Okay, training the personnel to do that testing for TB and putting people on INH. Today I won't be talking about this. I could talk a whole hour about the sample size calculation here, or maybe 2 hours if you guys want to stay around till noon. We could, but we won't do that today. I will spend a few slides talking about the order of entry, because we don't want to just start this piecemeal. We want to have a randomized entry, because it might be that we have all the big clinics or the well-to-do clientele start off first or last. Anything could happen. So we want to randomize the order of the clinics, and we want to actually avoid those kind of imbalances over time on those kinds of covariates as well. So this is a special case of a cluster randomized trial. The cluster are the clinic catchment areas or populations. Actually, I shouldn't even say area, because in HIV, sometimes people will come from the other side of town just so people won't see that they're HIV infected and so forth. So it's really the clinic populations themselves that are being randomized. This is why it's called a stepped wedge, because it looks like a bunch of steps. Every 2 months, 2 clinics are entering. Every 2 months, 2 more enter, and so forth. And by the time you finish, you got something that looks like a wedge. So instead of a stepped wedge, these are, as they come in, they're in those clinics. And this is a big distinction between clinics being in an intervention phase and individuals being in intervention phase. And we'll get onto this later. But it's a classic problem with cluster randomized trials. Things are happening at 2 levels, the cluster and the individual. So the clinics are coming in. They're in intervention period. They've got a control period for the ones who aren't in yet. And we had a lot of follow-up period as well later on. We didn't actually use all of that follow-up period for the main analysis. But we'll talk about the use of that intervention period in a little while. So the main analysis, these things are stepping in over 2.5 years. We have a 1.5 year follow-up after that. And we're worried there might be some time-related trends here. Now, this is a one-way crossover. You see people start in control for the clinic. Everyone starts in control, and then they go into intervention. So you could have a problem with if you don't account for exactly any kind of secular trends that are going on, seasonality and so forth, changes in the overall health system. And so the idea is basically to think, well, on each day, we're going to consider that a matched set, just like in the control group. Control group, they were the people at the clinics that hadn't been intervened in yet, so they didn't really get anything. Okay, I mean, they just went on as usual. So in the intervention group, I should say, they also had a retraining every year thereafter. So the idea is on each day, you condition on that day, and you see who was diagnosed with TB and who wasn't. Okay, it's a very basic approach. The only thing is we have to worry about later accounting for the correlation within clinic over time. So we just add a multiplier onto the front of right in here for another J sub I for each clinic to allow for the correlation that we have within clinic across time. So basically, to do this, we can just use a Cox-type regression model. Because the Cox model, you're going along in time, and every day you look and see who's the case and who's not, and you compare their covariates between the controls and the intervention. So it's a pretty simple analysis. We'll go on now to the randomization. Then we'll come back to the actual results. There's a lot of different ways you can step these people in 2 at a time. I should say the clinics. We're talking about entering the clinics. And we had a lot of covariates to worry about. And basically, we use this this equation here to balance the covariates. We wanted to balance the the covariate weighted months that people are in intervention and control status. And these are basically intervention months that a clinic is in and control months a clinic is in, weighted by covariance X. We wanted those to be within plus or minus a proportion, in a ratio, so that you're not too far from 1, so that the ratio of those covariate weighted months is not too different between the treatment condition and the control condition. So, as I said, we're not going to go through this in detail. You can read this at your leisure. Out of this will be on in a few days, I assume. We'll put this up. And we had to balance geographically. It was one of the covariates we had in there. This is basically Rio de Janeiro, different sizes of clinics and so forth. So these are the variables we used to, whether there's a DOTS program going on, and geography. We had some data on the baseline data on the clinic populations themselves. We figured that's going to be related to TB risk, so we'd want to constrain those to be, in this case, we wanted it to be plus or minus 10 percentage points of each other, the two conditions on the month-weighted covariates. There were so many of them, we had to randomly do it. Now, here's something you'll never do in any trial approved by FDA, but we took advantage of it. You randomize it according to the last 3 digits of this particular day's national lottery. That would never pass FDA, right? You never saw anyone do that. So we specified in advance. We're going to take the last 3 digits of that lottery and take the final 1,000 randomizations and take that. However, we made a serious mistake. It was a real problem, which is that a bunch of us went in and actually bought several tickets for that same day's lottery. Now, why is this a problem? Well, suppose we'd won. Okay, if we'd won, then everyone would think it would be rigged, right? If people think it's rigged, that's like the number one reason you do a randomization, to make people think it's not rigged. Anyway, it was a huge mistake, and as we said in the design paper, luckily we did not win. Okay, so first time we were happy not to win the lottery. Well, we had to have some method, right? And what we liked about it is that it puts the blame on the feds. Okay, the clinics, there's different clinic directors and so forth, and they say, why not my clinic? And that way it's a more objective way to say it's not rigged, or at least it's no more rigged than the Brazilian National Lottery. So so we could put it on them in a way. So that it's not extremely reproducible, but at least you could specify in advance and people could see. Anyway, but as I said, FDA is not likely to take it. All right, now, I just thought I'd bring up here the thing of design validity. Most people know about unbiasedness as a good characteristic to have in a randomization procedure. And unbiased design is basically the way it comes down to any unit has the same chance of getting the treatment as any other unit. Not 1 in 50 trialists, and I know I've surveyed them, not 1 in 50 trialists understands the flip side of that, which is what makes a valid design. And a valid design talks about how units are linked or not in terms of the randomization. And in a completely randomized design, it comes down to whether or not every pair of randomizations has the same probability of being in the same treatment arm. And this you can find this back in Fisher. He talks about plants on one side of the room or another. He's got it all down. But a couple of years ago, I ran into Rose Bailey, and she used to work at Rothamsted, and she's at Cambridge, and she was thrilled that people are paying attention again to this business. It's the problem is that if you have responses that are correlated, and if pairs of units tend to enter at the same time or not, then essentially you have fewer degrees of freedom than you think you have. You can imagine, if 2 units always are in the same treatment arm, well, gee, it's like they're completely linked. It's like instead of 2 units, you have 1 unit. But a lot of the systems out there for constraining the randomization, just like the one that we used, they they don't have a way to automatically account for that. So you have to look afterwards to see if you over-constrained or not. Anyway, we won't go into that whole thing, but it's basically why minimization itself is a bogus way to do things. It works though, so but it's a problem because no one ever looks for validity after the fact. Anyway, it's always a constant balance here. Okay, we check the validity. I told you I'd go through these fast. Okay, you can read 70,000, 5,000. Oh boy. Okay. Anyway, we had a validity matrix that shows you the number of times that these pairs had a chance to enter or not, and it's variable, but you know, it's not extreme. We've done some other simulations that make us think that anyway. And as it turned out, our final randomization with the National Lottery, we did pretty well. Our constraints were like plus or minus 10% for most of these, and we were well under that. So we got pretty good balance there. Richard Royal, one of our biostaff professors here, was fond of quoting I.J. Good, who said, you know, even though we strive for the lofty, beautiful ways to do things, make no mockery of honest ad hocery. And that's basically what we have here. But it works. We've got a balanced system and one that's also pretty close to being valid. To actually make something completely valid, it's virtually impossible. If you go and look at Bailey's work in JASA or Biometrica, you get into all this permutation group theory for the most simplest field trials. So it's just impossible in any given real situation that we run into to get something perfectly valid. But you can get something approximately valid that doesn't mess with your type 1 error very much. Okay. In this case, the last comment I'll make on the trio design here is that we did it because it was a politically acceptable and logistically feasible way to do it. You couldn't go out and train all the clinics at once, you know, and we could train 2 clinics at the time, you know, bring the personnel together. And politically, it seemed better because it wasn't like this was an intervention that we thought was going to hurt anyone, and it made sure that everyone got the intervention by the end. So it made it an acceptable design as well to the local authorities. Okay, so we'll start going into the results, but to see the results, we'll also talk about how we actually think about the data. So the main analysis goes through the first 4 years, which, as I said before, is about 2.5 years of stepping and 1.5 years of follow-up after that. And the way we're collecting the data is every once in a while going into these clinics and just abstracting the clinical records, which you can imagine is an awful lot of work. But it's surprising how well those records are actually kept, and we can keep finding the same ones over. And to lessen the burden, it's like every 10 months to 12 months, we go in and go through all the records and abstract everything since the last time we'd gone in. We could have waited till the end of the trial and just gone and gotten them, but it was safer and more convenient to just go almost every year, as it turned out, to abstract the records. But anyway, this is how we are counting them. Basically, we're looking at people who are either entering the clinics during our time or were there a couple years ahead of time, and we leave them in the denominator until censoring. The main outcome of TB, the very close one behind was TB or death, the combined endpoint. And we wanted to avoid people who came in just because they had TB. So we took out the first 60 days of TB cases after enrollment into a clinic. Let's see. Oh, we'll get into the stairs analysis here. Basically, we had another one which is sort of per protocol-like, but we didn't really have a protocol, so, it's not like a treatment trial. So the basic idea is that the analysis based on when the patients had the opportunity for the intervention. So we have, as I said, we have 2 layers in these cluster randomized trials. The clusters, the clinics, we're treating the analysis as an intent to treat. That is, the day the clinic is supposed to be intervened on and they start their training, that's when the clinic is entering intervention status. So the clinic is intent to treat. And that pretty much always happened. I think there's maybe one clinic where the intervention was delayed a little bit. But in general, that worked out pretty well. The The people, however, they're let's see. I should say that their designation of phase, whether in intervention or controlled, depends on when they show up at the clinic. So if they've gone for a year, they're supposed to be coming about 3 times a year to the ones who are in follow-up there. But sometimes it goes 6 months, 12 months without showing up. But the idea is to have it based on when they show up. So a person is in control phase, and I have about 3 slides that say this over and over, until the first time that they show up in a clinic, whether their clinic, which is already an intervention phase. And what that does is it mimics the long-term effectiveness of the intervention. If the intervention had always been there for the last 20 years, then, if you show up, you're getting the intervention. It doesn't penalize us for the fact that we're trying something new. So it mimics that long-term action, you could say. All right, so people, their control and intervention phase is different from the clinics. We'll see that. I got a picture of it too. Okay, I already said the Cox model with the random effects, and so that's a binary covariate for each individual that tracks when that individual is going from control to intervention phase. And it's basically, it really is the same thing as looking at it globally at the clinic level and looking at the proportion of individuals who've shown up for the intervention phase. So it's like a partial allocation covariate is what it turns out to be, which Hussey and Hughes had proposed some time ago. Now, we had some slight problems. We weren't exactly sure when all the clinic visits were for people coming in. But so what we were looking at was for meaningful clinic visits, to know when to start someone's time in an intervention phase. And so if there was a record of, they're supposed to have their CD4 done every time. So if there's something in that clinic record, or one of the, I don't think we listed, maybe 8 conditions, then we said, well, they had a meaningful contact with the clinic, and that's when we can start them. Okay. All right. Again, if TB is diagnosed before a patient's first clinic visit during the clinic's intervention phase, that is counted against the control phase. Otherwise, it's against the intervention phase. I just want to emphasize how we're looking at this in the Cox model. We've got these vertical time slices. Here I have one slice for every step. It's actually a slice for every single day. Just to keep in mind how that perfectly is accounting for any secular trend. Because on any day, you're comparing intervention to control, and then we just combine things across time. Okay? All right. Now, clinics tend to treat. Individual, I said I had about 3 slides on this, right? No one's forgotten it. Okay? It might be 4. Okay? Dynamic cord, yeah, so there's people coming in. We had some problems with transferring and so forth. But here's an example, just to really fix it in your mind. So here's a study timeline here. And here we have now on top, this is a clinic timeline. So particular clinic, actually it's starting off in back here, in intervention and so forth. So in an individual, I'm sorry. This is to track the individual. This is a different picture, I think. Yeah. We're taking just one clinic. We're thinking of a clinic that comes in and their training is right here. We don't have a line for the clinic. One clinic that's training is right here. An individual here is in control until the first visit they have, which is here, after the clinic came in. So they're in control phase until they show up in the clinic, which is already in intervention phase. So anything that happens here is in control, and here's an intervention. So it's a little bit different. Again, it's 2 levels, and it took us forever to realize this, probably 2 or 3 years. Anyway, so there was, it took us a long time. Let's put it that way. All right. There was, however, a lot of patients who weren't showing up, and so we were a little bit worried about them because they'd been in there 2003, 2005, a couple years beforehand, and then they never showed up, and, well, were they ever around at all? So we invented this thing we called the Stayers analysis, that we keep people in until they'd gone a year without any contact with the clinic. It's not a per protocol, but it's to make sure we're looking at people who actually have a chance to get to the clinic and get its intervention. Okay. So, and those people who are so-called lost, they had very little information on TB. We had all sorts of systems. They were registered, a big TB registry in the whole city, the death registry, and so we were trying to get extra events from those as well as from the clinical records, and actually did get some extra ones, but not many on the ones who were truly lost. They were just adding a lot of denominator time without many numerator events. So we made this sort of adherers analysis. It's not much of an adhesion. They're supposed to come in 3 times a year, and we've said, well, if they just came once, then we'll keep them. If they went for past 12 months, then they were censored at 12 months after their last determination or last visits. Anyway, it's just this is a secondary analysis, of course. So here's all the data. Not that you can see anything from it, but that is, there's so much data, and then the effect, as it turned out, we'll see, 10, 20% effect isn't enough that you can actually see it from this picture. But one reason I wanted this picture made was just to help with diagnosis of the data, just make sure there wasn't too many funny things going on, the things we thought were going on were there, and so forth. And so here's the time here. And now these are now just the order of entry of the clinics. So here's our wedge here. 2 clinics coming in here, 2 here, 2 here. So each line is the experience of a clinic. And these are all the TB cases in the calendar time when they happened, when they were diagnosed. There's 475 of them on that picture. For those of you, the human brain can comprehend maybe 100 objects. This is about 475, in terms of understanding how many there are. And the blue are the ones that count against the control, and the red against the intervention. And so we see that even after the intervention, we still have, in a clinic, some cases that are counting as being cases that occurred during the control phase. Again, that's because they had not come in yet and had the chance yet to be individually in the intervention. They would have if if this program had been going on for 20 years, when they enrolled, they would have been in there from the start. But if they hadn't had the opportunity yet even to get that intervention. So they're still counting for controls. And what this does is, in an ordinary stepped wedge trial, you're finished right here. After that, everyone is in intervention phase, and there's no more contrast between intervention and control. There's no more information you can gain on the treatment effect. Not when you're accounting perfectly for time. There is some more information in terms of after minus before. So in a stepped wedge design, you actually have information both vertically and horizontally, before and after. So you do have that added. But here, these cases are still contributing to that vertical contrast that gives us information on the effect. And that is going on up till near the end. So. Most of them, actually, because as you're right, they had to get diagnosed, and so they had to come in to get diagnosed, but then they didn't have time to really get the intervention. So almost all of these, I think, were through the registry, as it turns out, because they couldn't have come in several times beforehand, because then they would have been in the intervention phase themselves, right? So, yeah. Yeah, because if they come in, because they've already been in the clinic, the 60 days thing just corresponds to the people who have never been in the clinic before. So they're here, they've been in the system, okay? The person is in the system, and then they show up basically with TB. It's a 60 days thing is only applying to those who had never been in the system before. If they just show up off the street and they say, oh, I've got TB, that's why we're taking out the first 60 days. But these folks have been in there for a while. They come in, and it is their first visit in intervention, and they're getting diagnosed there. So some of them are getting diagnosed there. A bunch of them were found outside. See? But it's not their first. I guess they would turn intervention the day after that first visit, but then it's too late because they're already censored. Okay? All right. It took us about 3 years to figure that out. But it's the distinction between those that are coming in and those who have been in the system already. So they've been in there, and then they show up with their TB. And so they're counted against this because they hadn't been there before. The day after if we were looking at repeated TB, then they would have been in intervention the day after. So we're just looking at the first event, which is basically all there was for all of them. I don't know. I might have had a couple of things. Anyway, we've gone to some of the results here. This is sort of a before-after thing, a grouped before and after. Control, what happened in control phase versus intervention phase for the individuals. And if we look at the cases here, let's say 475. By golly, it adds up. Okay. You do actually see a bit of a reduction in the rate before and after, but we can't just be satisfied with analysis like that because maybe there was just less TB later on in time, in several years. So we can't be happy with just this before-after look in the rates, but it makes us feel good anyway. Okay? All right, and here it was for TB or death. Same thing, we see some reduction there. Okay, here's an analysis we're not showing anybody. This is a pure vertical analysis that you see done in some stepped wedge trials where they're just looking within each step what's happening. And within every 2-month step, just for the heck of it, I said, well, let's calculate the rates in that 2-month step. Of course, it couldn't possibly have any effect because in 2 months, you couldn't, by the time most of the 2 months are over, well, you might have a couple people on isoniazid. There's just not enough time to get them tested and on INH. Anyway, and as we see, 1 was for intervention, 0 for control. Almost every 2-month step, actually the intervention clinics had higher rates than the control clinics. 1, 0, 1, 0, 0, 1 the other way, 1, 0, 1, 0, 1, 0, 1, 0. It looks like the intervention is hurting. But what we think is happening is just, we intervene in those clinics and paying more attention to TB, it's getting diagnosed more. Okay, just in the first 2 months. It's impossible to really have any effect. But it's the kind of thing you would expect to happen, that they're finding a little bit more TB there. This is a pure vertical analysis that often you see in stepped wedge trials. But you have to be careful to make sure that, you want to design your stepped wedge in general so that all the action takes place in that step if you're going to analyze it purely within the step, which can happen, say, in a hospital infection control study where right from day one, everyone is getting a new wipe or something, and then you can stop it whenever you want. You can see right away within 3 days whether the person get a post-op infection or not. Here, these things taking months, not years. We also thought we'd look, because we were doing this Cox model to avoid problems with secular trend, we thought we'd look and see what the secular trend was. One easy way to do that is just to take the last clinics to enter the intervention phase. Remember, these are randomized, and just see in them over time, they're in control almost the whole time, just see what their rates were. So this is a smooth hazard of TB over time for those last clinics. In fact, it looked like it was going up a bit and then down over time. And we looked at this. We were down in Rio at the time when one of our epidemiologists projected this on the wall. And another epidemiologist looked out the window while we were there. And she said, "Look at that." What do you think she saw out the window? Sugarloaf. She said, "Yeah, it's just exactly the same shape, a little nose and everything." And it was one of those cosmic trial moments. Okay? And I've acknowledged Catherine back here who fit the line as best we could. The scale wasn't quite right, but it had the right morphology anyway. So that made us feel good that the data were probably correct. And there seemed to be a real trend there. It's just another way to plot the data. When you have studies with new designs you haven't been with, you want to plot the data every way you can just to see if anything pops out. This was looking now at this is the difference after minus before. Okay, so sorry, for each clinic, I'm taking the TB rate after intervention minus before. These were the rate differences, and this is by order of entry into there. So just to see if there's anything weird going on with the order of entry into the study. In fact, there might have been a little bit. It looked like there was more variance for those who entered at the start. And we actually, but more than you'd think. You would expect a little bit more variability just because, they've collected more Poisson events over time. But we didn't see anything particularly that bothered us. There is an outlier. This is one small clinic that had 2 TB cases. It was a very small clinic, and so it's an outlier in all of our plots. But, you just want to see if anything funny is going on over time again. Just a sort of diagnostic plot. Anyway, so here it is with the actual Cox-type model regressions. The absolute main effect was only 13%. Pretty big p-value. But TB or death, that was looking pretty good. They got 24% reduction. It's a little bit better. And if you look in the stairs, then we get the effects of 50%. I mean, we know isoniazid works, so it's not too surprising. And it's just that we found out when we were looking at the data, it's taking a lot of time to get people, even in the intervention clinics, tested and on the isoniazid and so forth. And there's a lot of variability over time too. And so the actual number one endpoint didn't quite make it, but this one was close behind. And a lot of these studies, we combined TB or death anyway. We had another TB treatment study in South Africa, and halfway through this trial, the DSMB said, well, you've got to have a combined endpoint for your primary endpoint. And we changed it just because there's a worry about too many people just leaving and dying, and then we never know if they have TB or not. In this case, it wasn't so bad because if people stayed in Rio, but if they then left Rio and died, and then we find out they died later somehow, then they could have had TB first and then died. So as I said, this got published, which means we can actually show the results. And so I'm going to conclude because I know people want to ask some questions about stepped wedges probably. That's why half of you are showing up. I don't know why my enemies are showing up. But anyway, there's a lot of problems you can have with stepped wedges. We didn't look at this today. You can have a significant loss of power. I'll just say something about that. In fact, if there's zero within clinic correlation, then you have the design effect for the stepped wedge itself. The wedge itself is about 1.5. That's the limiting factor. The more steps, the more design effect, and it maxes out at 1.5. That is, you need 50% more clinics than you thought you were going to need. However, if the more correlation is, that the more that design effect comes down, because then you can retrieve some of that information because you have the before-after comparison, and accounting for that correlation sort of ties those together, just like in a paired t-test. And you can get to the point where it's actually more efficient, but it means you have to rely on assuming there's going to be a certain amount of correlation. Anyway, it's risky. There's a balancing of the randomization. We did sort of an ad hoc way. Handling the lag in the implementation or in the effects of the intervention. And actually, we sort of ended up, in a way, doing these diagonal analyses, if you will, because people are being compared later on in time to where their clinic was. They're not at the same place where the clinic was earlier on. And so they can be in control until they're showing up that first time. And there still can be other problems. We have, for the main effect of time, where we handled that, but there may be other secular trends that do linger in terms of interactions with time. And you can have problems with, especially if you just have, ours was more dynamic, but some situations you might have just a fixed cohort of people, and then they're aging, and there's only so many ways you can account for that, because then some people have been exposed, it depends on the intervention, they've been exposed to the intervention for 6 months, some have had 9 months. The whole thing can be a mess very quickly. So don't try this alone at home. Use a parallel design if at all possible. People often think of, you see people write stuff like, oh, stepped wedge is more ethical because everyone gets it at the end. Well, it's more ethical to get the answer quickly, if you can, in a parallel design. And then if it works, then just give it to people. If you have the money to do it, you can do it. We're doing that with the trial in Zimbabwe. We have it, given some people toilets now. At the end of the trial, in fact, the name of their treatment arm is toilets later. Means they're going to get it. Okay, these are the enhanced VIP latrines. Anyway, so they're going to get it. So anyway, I'll end there with just saying, watch out for these stepped wedges. These things are dangerous. Okay. So we better have some questions here. So who's got the questions here? Okay. Well, since you gave me such a nice introduction, we'll call on you first. Yeah, Patty. Oh, a typical one? Well, we've accounted for time as best we can, and basically you take that those hazard ratios, and we have a confidence interval for them, and you just throw that in, and just hope that with any other trial that things have been properly adjusted for. In this case, we handled the main problem with time. There could be some other problems, of course, but I wouldn't give it any weight less than what your confidence interval comes. It's just that the main problem is that you have to make sure in your meta-analysis that you're estimating the same parameter. This is the biggest mistake people make in meta-analyses. They're not estimating the same parameter in the different trials. Here, there were 10 different things we could have done in the definition, as we even talked about today, is the when person or the clinic starts an intervention and control phase. Really, there's all sorts of different things we could have done. And each one estimates a slightly different parameter. And so what's the chance that it's estimating the same parameter in the other trials? I think not that much. So if you're close, if you're just trying to estimate, a proof of concept, was there any effect over all the trials, then yeah, you can combine them. But this is the main problem with a lot of meta-analyses. There's just not the same parameter. So that's the thing you got to watch out for. Yes? Consent? No, because it's already on the books. It's already supposed to be getting this treatment. So there's no one consented for anything, except the implicit consent of the people actually who are implementing the study, all the clinic personnel and so forth. But no, there's nothing like that. No one got any consent. You know of, do you? No. And then and then we were working with the lead author and a couple of the others are actually the government personnel who are running these. They're in charge of all of HIV in Rio. So we've got the consent of the whole government, and it's their facilities and everything. So that was essentially built in from the start. Clinic level, from the director. Okay. Jonathan Golub, star of the study, among others. Okay. Yeah. Oh yeah, yeah. We spent a lot of time figuring that. We tracked them, I think, for the first 2 clinic changes, was it, and then gave up? Is that what it was? Yeah, but once they were in intervention, you see, that's the thing. If they were intervention, that's who's right. So he's got it. Once you're in intervention, you say, well, you had your opportunity to get it, even if you switch back within a month. So we're going to keep you in intervention. So, but it was there wasn't as much mobility as I thought there would be, actually. But it was a pain tracking people to see how many people are going to second and third clinics. I had a question. Mm-hmm. Actually, for the for all the analyses there, we're doing intent to treat with the clinic level, and there's very little difference as to whether you treat the clinics as intent to treat or not, just because there's really just that one clinic which was a little bit later. Now, for the individuals, so we did have an analysis where we looked at them and switched them at the same time as the clinics. I think that's what you mean. So if we treat the individuals with the intent to treat too, and as I recall, we had very little effect for the first one, but still some effect for the stayers. That's what I remember. So it's a different parameter you're estimating. And when it took us a couple of years into this study before we realized that's not the one we want to estimate. I don't know why it took so long, but we did. Yeah, that's good. Yeah. Pretty much. I mean, there was a political aspect too, because it sounded better that everyone gets the intervention. And it guarantees it as well. Because it's built in, but there's so many problems in the analysis. You lose. Trying to explain this to people in a journal article or anywhere, it's almost impossible, as you can see. And so it really is. It is if you're forced into it. You can do it. There are other ways you can do things logistically, though. And we had another case finding study in Brazil, which is done differently, where it was a multiple baseline study, but with every pair of areas that came in, one was treatment and one is control. So it's paired, and that's probably a better way to do it if you're logistically constrained. So you could have with every pair that came in, what was it? It was a leaflet or door-to-door. A case-finding trial. And so they came in with pairs, and one is control, and one is active control, and then the intervention. So that way, at any time within that pair, you have that paired comparison, and that's maybe a better way to do it. It still has problems, though, because there's the maturation effects, because some people at the end of the study had a lot more time than others, and you have to think about how long that effect had a different kind of treatment effect. Yeah.
So, do you think in this intervention, was there sort of a lag time before the intervention took hold? Because you're talking about a training, and then they have to actually go and implement and so on. And so, is there a way to sort of adjust this analysis in some sense to say, there's some lag time after the start of the intervention where you don't really think that there'll be an impact?
Yeah. And we could have done that. Each person could have added the time-varying covariate that automatically waits 8 months after their clinic comes in, and then that person, all the people in that clinic get in there. All the clinic comes in there. You can do that.
Sort of a bit smooth because you sort of think it's getting better and better
over
some time.
Well, that's you can do that, and that's essentially what we did with the individual patients. It's really the same thing as if you're saying at what point in time, what proportion of the people have had the first contact. So it's the same thing. Is that you can have that continuous. At the clinic level, it's a continuous measure, going from 0 to 1 essentially. And so you can do that. And we had a trouble like this with another Zambia trial. And the problem is to find an independent measure that's not part of the outcome that can track that intervention, because you're not, the intervention doesn't happen on day one. There isn't a lag. In this case, we were able to figure out what that lag was for an individual, or what it could be for the individual. Because in any clinic, if you ever come in several times a year, even at the best, it's going to be sometime months before they're all come in to even get intervened upon. So you don't want to count all the TB that's happening against that, because that doesn't tell you what would happen in the long run in this population. Mm-hmm. Oh boy, we're getting all sorts of folks here. Okay, we'll start up here and go back. Yes. No, and that's one of those graphs sort of got at that. If anything, it was the other way around. Um, and it's really, these are only like 29 out of 54, 56 clinics in the whole area. And there's an awful lot of TB going on among people who don't have HIV. So there's really no way we're going to have any kind of indirect effects, if that's what you're getting at, in terms of stamping out TB in the population. And so, and in fact, even in the clinics, overall, it's a pretty modest effect, just because there's just so much TB going on. Mm-hmm. Going on there, Marie. Yeah. That was for the last 5 because they stayed in control of the whole thing. No. Yeah. Well, we could but then they would all have much shorter times. Like the people who entered the clinics that started right away, we could look at that. This is without a treatment. But if a clinic started their treatment right on, and they have a very short time, we could have added up all those little times there. But then, I was just trying to do this quickly. And that could have affected it a little bit. But once they're in treatment, also the treatment it's not treatment right away, as we were saying. It's variable treatment over time. So you had to get the pristine clinics most of the way through there. Yeah, it was only based on 5, and there is some variability. But yeah, that's a good point. Yeah. Mm-hmm. Mm. Oh yeah, that could happen. The trainers might get tired, right? You just have one set of trainers. Maybe at the end they're either a lot better and more efficient at giving the training. They know what works or not, or they just become blasé about it. It can work either way. In this case, we kept going back a year later and retraining to sort of keep a constant level of awareness among all the clinic personnel. But that had some little boluses too. You could, yeah, you're right. That can be a problem. And that's why it's probably better to pair them up at least at that point in time, and then you can look for interaction over time to see if there's some drift in the training. But yeah, that's the thing with the health services thing, as opposed to say a study where you're giving everyone the same drug over time. Hopefully that drug is stable and it's Any check we could make on that? I don't think so. We had the regular training reports just to make sure they were happening and people that attended and this and that, but I don't know. The quality of the training. Yeah. Yeah. In fact, some clinics, probably the ones that were trained even before the retraining, everyone might have left. That could happen. Hopefully, they trained the new people who came in to do it, but. But this, this is mimicking real life. That happens in real life, where there's personnel change. So, yeah. I never heard anything about a stock. Yeah, no stockouts. Yeah. Good question. Oh yeah. Oh yeah, you're right next to me. Okay.
Yeah. Since you mentioned a couple of times that it took the study team a couple of years to figure out
Yeah.
the parameter you're interested in estimating.
Yeah, we kept changing the.
Wondering what was the parameter that the study was powered for at the beginning?
Yeah. Well, that's what I said. That was 2 more hours talk goes into the sample size. It was still on TB, but we were, and the idea was. That once you intervene in a clinic, that's when the effect was going to take place. And so it was based on the rates that we had from looking at a sample of clinics from before. And we said, well, when that clinic comes in, we're going to start counting TB cases. And so it was not quite the same parameter as what we ended up with. Now, after the fact, sample size, I mean, I don't know if I should, now that I'm being recorded, I should say it, but I don't even think you need to report it in a consort. The consort people would say you have to report sample size calculations. I don't even believe that. After the fact, you've got the data. You want to do sample size calculations, make sure you're going to get what you want. Yeah. I don't know, people like to see it. So at least it gives you an idea of how people were thinking about this study. In this case, you would have seen that we were thinking about it a little differently, that when you intervene in a clinic, that intervention takes effect. But it doesn't, because the people aren't there. The clinic got trained, but the people haven't come in yet. And so it was a bit different. On the other hand, we had the sample size calculation was only done for that first 2.5-year period. We only counted that time during those 2.5 years. And we had a lot of that extra time afterwards that we ended up using both contrast in the time and before and after. So we had actually more information, and that sort of made up for it, if you will. And actually, our coefficient of variation was spot on. Oh, it's incredible.
Is there any seasonality to TB, or is there seasonality to when people visit clinics?
In Rio, I don't think so. There's no snow keeping people from getting to the clinic like they have here. There's no real seasonality in TB. Probably anywhere in the world, is there? I mean, if you
some places in the world,
yeah, or so where people may spend more time inside and you have more close contact.
But if you have seasonality with an outcome, doesn't that sort of complicate?
Well, not so bad here because again, here we're the Cox model is saying everyone on that day, who has it, who doesn't. So that really accounts for that.
When you do this pre-post part of that.
Yeah, if you only did pre-post, hey, you'd be in big trouble.
But so if you're combining kind of the pre-post with the cross-sectional to get an estimate, then you have to deal with that issue.
Yeah, but it's not so bad because there's different ways people adjust for time, and sometimes they'll just put in a bunch of dummy variables for a different time, and so they may not completely account for that seasonality then. Because if the dummy variables may be too big, they might have several intervals they've combined. So you can have some of that residual confounding. But here, I think that wasn't a problem. Well, one more, just for the road.
So if you have a time stamp, then along with it, over the time, study has proven twice.
Yeah.
Do you think over the time we may not see that improvement? It's just not because of our pain, because more people are treating. You might just get things from
Oh yeah, no, that's what we were saying is that there can be that effect, and so you could put in an interaction term. I haven't done this. You had another interaction term that has time by treatment, and it's just that at the end there, you're right, you don't have much contrast anymore, and so if something's happening in the last few months, almost everyone is in intervention. It's like a matched case control study. We have 10 to 1. You really don't have much contrast anymore. So at the end, you don't have much information to tell if something's changing at the end. Okay, well, maybe we ought to stop there. Okay, well, thanks for coming out. Okay. All right.